to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 88th episode of the Weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is John Perkins and I've been in the bioanalytical LC-MS space for around 30 years. I'm here with my co-host Dominic Warrino.
Hi, my name is Dominic Warrino, and similar to John, I've been doing this for about 30 years.
Dominic and I are members of the growing scientific advisory team at KCAS Bio. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS Bio's services. Today we're here with a number of guests: David Ambrose, Jessica Pham, Bryan Parmentier, Ravna Veeramatchaneni and Don Duffield for an action-packed main topic discussion. We're thrilled to have you listening to the 88th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources and then a focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics and we'd be happy to discuss something that you want us to cover. So again, we're fired up to have you here and we are looking forward to a fun episode today. To kick us off for podcast number 88, Dom is going to go over today's podcast topic a bit before we jump into the news and resources section. Dom.
Yeah, for those that are new, we always start with some news and resources. We'll go over a few topics in the latest and greatest in the medical research field. They're topics of our choosing. And then for today, as John mentioned, we have a whole list of guests, and we're going to go over reflections on WRIB in 2025. So, John, let's just get right into it. Let's start with the news and notes. What we got up first?
So we've got a fair amount of basic research to talk about. It's always nice to see what might be coming to us in the long distant future. First one is newfound compound kills multi-drug resistant fungi. Or fungi, whatever. So this is out of the China Pharmaceutical University in Nanjing have identified a bacterial compound that can kill infectious fungi even if they're resistant to antifungals. Discovery of the compound called mandamycin was reported in Nature, and they basically grew a suite of drug-resistant fungi in broth, exposed them to the compound, and they found that this compound mandamycin killed species of Candida, Cryptococcus neoformans, and Aspergillus fumigatus, according to the team. And they basically shown that they could then translate this to mice treated infected with Candida auris. And the interesting thing about this is these are all fungi that have proven resistant, but this compound has a fairly unique mechanism. It's a macrolide antibiotic, and it binds to phospholipids in the fungal cell's membrane, causing potassium to leak out the cell and ultimately collapsing the membrane. So there are other macrolides out there, but this is really quite unique in the way it's done. And the reason why I highlighted this is it's basically the group were in terms of how they identified this compound, they basically looked at genomes of 316,123 bacteria from the National Institute of Health's National Center for Biotechnology Information. Because what they were interested in is that bacteria living in the same environment as these kind of fungi, they're competing for resources, so they actually often come up with compounds to snuff out the fungi so they can succeed. And that was the path to identifying this compound that, long term could have some useful application. I'll pass it back to you.
Every time you say fun guy, I think, yeah, I'm a fun guy, John, but
I'm way too old to be a fun guy.
This is really, John, it's really fascinating stuff. We love to talk about, kind of less immuno-oncology, more of things that can help global health, right? This is a big impact on global health. And really one of the things that I think really resonated when I was looking through this article is they say it's like hacking 2 billion years of archived files from the Global Bacterial Military Central Command and placing them in chronological order. I thought that was a great line, John. So really you talked about this 316,000 different types of bacteria. And then the mechanism of action of this is what's really unique, right? So all other like kind of antibiotics that are on the market for dealing with yeast, they discovered something brand new, right? This is really fast. It's amazing, and hats off to these scientists. I don't know. I don't have much else to add other than like I don't. It's not my jam, so to speak. I've never loved microbiology. You talked about broths and tryptocase soy agars and stabs and all these things that I've got some experience doing, but it wasn't my passion to sit here and mull over 316,000 different bacteria types and to kind of, to me, it seems like they really, I can't tell if they got lucky or if it was hard work, John, and maybe they go hand in hand.
I think it was hard work
to get this mechanism, right? And the importance of this is like, none of these bugs are resistant to it, right? They pounded the animals and stuff, and none of them became resistant.
So the choice of fungi had been resistant to between 2 and 4 existing drugs, and so these are, I mean, just to go talk into the background of what they were looking at. Candida are yeast that normally occur in parts of the body, but they can cause infections called candidiasis, where they stray beyond their normal bounds. Serious invasive infection can occur if the fungi reach the bloodstream, heart, or skull, which is especially a concern for immunocompromised patients. And then Cryptococcus neoformans form spores that can cause lung or brain infections when breathed in, and Aspergillus fumigatus, a kind of mold, can also cause respiratory infections. So, it's just they're nasty if they get to the right people, if you see what I mean.
This kills people, though. All those things you just described are deadly to immunocompromised and the elderly as well, the senescents.
Sure. Yeah, so that's why I always like to see these sort of things, and it's again, it's not necessarily exciting scientific developments, but it could have big implications for people in the long term. So
it made it to Nature, right? Nature's a great publication.
Exactly. Yes. So let's move on.
Yeah.
This might excite you more.
It does.
So, edited T cells allow gluten-sensitive mice to eat grains. So, according to the article, there are 80 million people worldwide with celiac disease, and so, right now the only real treatment available is to just cut past it and bread out their lives. But this is a group in Lausanne University Hospital and University of Lausanne in Switzerland, came up with a cell therapy that was able to tamp down the immune system's overreaction to gluten in mice. So obviously early stage research again, but showing potential where we could possibly go with here. So basically gluten triggers an intense immune response in the intestines that leads to gastrointestinal symptoms such as stomachache and diarrhea. And again, no approved treatments, just stop eating gluten. So basically this team wanted to look at regulatory T cells to try and hit the brakes on that overreactive anti-gluten immune system response in celiacs. And these regulatory T cells have been applied in other diseases and other clinical trials, and so they thought celiac was a good candidate for this kind of approach because more than 90% of patients all have the same genetic variant. So it's like with that high similarity, they could use T cell receptors to redirect the specificity of the cells. I'll pass it back to you.
Yeah, it's. I didn't realize that it's the DQ 2.5 variant of HLA. That's your class 2, and 90% of them are DQ 2.5. So that's like, to me, it's causative, right? And so then I didn't realize celiac had that high an incident of that variation in their HLA DQ, but that became the target, right? So CAR T cells, cell-mediated immune cells, genetically modified cells, but now they call them immune effector cells. So they took and this immune effector cell therapy, it was autologous, so cell therapy.
Yep.
What they did is they just re-engineered that DQ 2.5 to actually kind of they generated a suppressor T cell CAR T. And that's where they're looking at it in autoimmunity and other rare diseases. This is the first I'd heard about it being used in celiac disease. It's just brilliant, John. I'm blown away.
Yeah.
It's unbelievable the way they had tackled this, because when you think of T cells, they're usually like attacking or helping or killing or whatever. but there is a whole subset of regulatory and suppressor Ts. And so in this instance, brilliantly enough, they've generated suppressor T cells that are basically dampening down. And again, the key behind all of this is that mutated DQ2.5.
Yeah.
So they're able to kind of target that, and seems like they've alleviated this. And one real important note here is that an autologous therapy is incredibly expensive. There are 80 million? Is that what I read? Something like that. There's 80 million people with celiac disease.
Yeah. Yeah.
So getting autologous therapy for that, John, that's not a feasible. But they're looking towards these allogenic off-the-shelf approaches, but those are going to take some time. But really, big breakthrough here.
So just for people who aren't aware, and I wasn't necessarily aware, autologous means that you've taken cells from the patient, you adapt those, and then re-administer them to the patient. With the allogeneic approach, the edited cells come from a different person, so that's how you reduce the cost. Yeah, just to talk about the mechanism, basically the team edited the T cell receptors of regulatory T cells, so they were also activated by gluten, triggering them to perform their function of calming down effector T cells, which are the T cells that aggressively react to gluten so basically, reduce that reaction. It's really clever.
Yeah, they took advantage. I don't know how recently they found out that it was a DQ 2.5 variant, but the moment they learned that became the ability to. I don't think you can cure celiacs, right? You said it best. You have to alter your diet. There's some other. I think TNF alphas help a little bit, and there's been some really wonky things where people do like swapping out of like your microbiome, right? They try to do some things there. None of that really kind of took off, but this, on the other hand, seems promising. Of course, thanks for clarifying the autologous. What that means is it's very costly. Can't imagine we'd be able to mainstream treat 80 million people with autologous, but using an allogenic form, and those are fascinating things. And maybe we save that for a whole nother podcast. But those have really made some advancements since the first CAR T cell, which was approved back in maybe 2015 or 16, I think was the first approval. So 10 years later, we're starting to look towards getting them like cells in a bottle that you can someday, John, our grandchildren will be able to walk into Walgreens and treat themselves with an allogenic mixture to prevent them from having cancers. That might be what's maybe it's our grandchildren's grandchildren.
Yeah, so the head researcher made the point of like autologous is very, very costly, and so they want to move towards allogeneic, but an off-the-shelf approach, but that's going to take time. So it's, as we talk about so often, it's the baby steps to getting something in the future. But
the other thing is it becomes a target for like an AAV approach, right? More some sort of gene therapy to modify the regulatory cells transform into suppressors, right? That's the mechanism here. Okay, John, we got a big crowd with us. Let's move on to our last
one more. This is quite close to my heart. This one after very miserable experience in Venice this time last year. Drug for rare disease turns human blood into mosquito poison. I so wish I had this to hand a year ago. So basically, scientists have a radical new plan for controlling mosquito numbers and fighting malaria, lacing human blood with the drug that's poisonous for the insects, so sucking on this blood marks their last meal. And this is work done by the Liverpool School of Tropical Medicine in the UK. And they were looking at nitazoxanide. It's actually used and approved for treating certain rare inherited diseases, and it works by blocking production of a specific protein which leads to reduced toxic disease byproducts in the human body. But when mosquitoes drink blood with nitazoxanide, they quickly die. This is all early proof of concept stuff, but they were just interested in saying, what could they do to basically stop blood sucking insects. It's basically the quote from the microbiologist Lee Haynes. One way to stop the spread of diseases transmitted by insects is to make the blood of animals and humans toxic to these blood-feeding insects. Now, there are precedents to this. Ivermectin's actually been used with I think it's sheep and cattle. But what they've seen with ivermectin is actually it's more it affects other organisms, so
Yeah.
it's not just mosquitoes. Whereas this nitazinone,
And it
at least
clears
to
quickly
date,
too, right, John?
yeah,
It's toxic and it clears a little bit
yeah.
faster. Go on, yeah.
So the nitazenones actually seems to be more specific to the mosquitoes, so it's less of a broad effect. So they don't know about in terms of long-term resistance, things like that. But again, it's another researcher talked about, and we thought we want to go down this route. Nitazinone had to perform better than ivermectin. Indeed, nitazinone performance was fantastic. So I just, yeah, malaria is one of those things where I think I think there's vaccines out to it, and there's other treatments, but it's just such a ubiquitous disease in the tropics that you know any mechanism that you can explore that might help control malaria has got to be a useful step forward.
Yeah, it's a big fear for anyone traveling, right, John? It definitely is. If you travel to Africa, you've got to be careful.
Yep.
Malaria is so prevalent. Just a quick, study malaria a little bit. Why it's so evasive? It goes into like it harvests itself, and the cycle of it's like 5 different stages throughout your own body. So it's so hard to treat it. That's why it's difficult to treat malaria, because it just kind of takes over, and the cycle of it is just evasive of the immune system. And then I got to give a shout out to the Liverpool School of Tropical Medicine. That's because Liverpool's going to win the English Premier League Cup, John. So I want to give Liverpool a shout out. But no, this is another story that's really incredibly, to me, it's great reverse engineering, right? You had avectomycin on the market there. They discover this new drug, and what does it do? It actually like, what do they call it? Like killing juice or something like that of the bug, right? Of malaria. It's unbelievable how the thought of how, I guess, again, because we don't study this on a day-to-day basis, I find this incredibly fascinating with people that are studying infectious diseases and these global kind of, I don't know, epidemics, I guess, or global kind of, diseases. It's just, to me, it's so clever the way they said, hey, let's not try to treat a single stage. Let's treat the animal or person with something that the moment the animal, the bug tries to, presumably the infected insect, right?
Yep.
Tries to sample blood, it just kills it. So I mean, it's incredible. I don't know if I articulated that well, but to me, it's like, not only did they make it less neurotoxic, they found something that could be well tolerated in humans and mammals, and became incredibly effective at killing the bug itself. Just great science there, John. I wonder, like you said, we don't know if there's any side effects just yet, but from what this looks very, very promising. And I mean, to me, it's like, if I'm ever going over to Africa, which is on my bucket list, I'd love to go to a safari. I want some of this, right? That
absolutely.
I want to take, even if it's not fully approved yet. But like when they said fed on 3 people who were taking this nitrosone 9.
Yeah, it was yes, exactly.
The insects died within 12 hours, right? So it, like, in theory, it could like knock out all the malaria in the bugs. Although, is that really possible, right? But that's kind of the rub here. It's fascinating stuff.
Well, I suppose it takes out a subset, but at least it takes out a subset. It's like, so the researchers tested the effects of nitazinone-filled blood on mosquitoes, as well as using certain models to figure out the impact of different doses on simulated populations. They found the drug was effective at killing mosquitoes at all ages, including the older insects that are more likely to be carrying malaria. So again, it's the real practical application of this kind of approach. Yeah. And you're right. It was about the 3 people, mosquitoes fed on the blood of 3 people who were already taking nitazinone to treat a genetic disorder, and the insects died within 12 hours. So I mean, honestly, if you're going on a trip and all you have to do is take a short course of pills to help keep it under control, that would honestly be great.
Appear not to have much side effect. And by the way, malaria is still responsible for 500,000 deaths.
Oh sure, yeah, exactly. It's no trivial problem.
Yeah. Well, and then it was in Science of Translational Medicine, another cool little.
Yep.
So, John, any last parting thoughts before we jump into our main agenda here?
Let's go into the main agenda topic, and this is going to be this is going to be an interesting. This is something we've never done before.
Unprecedented, John.
Yes,
Unprecedented here on the Weekly Bioanalysis.
We're pushing the boundaries as hard as we can go. Obviously, last week was WRIB, the big bioanalytical meeting for the US. We had a number of scientists presenting there, and obviously a lot of attendees from the company. And we thought it might be good to, again, try and do something different, have people talk about what we did in terms of the science that we were presenting, because we want to be out there talking about what we're doing to solve real world problems, and then just a general discussion of the meeting as a whole. So with that in mind, we have a number of guests, and we're going to go through. I'll go through the list as they are in front of me. We had Don Duffield was on the last podcast. Don, do you want to say hello? I mean,
yeah, hi. Yeah, it was nice to join the last one. We talked about some SciEx stuff, and this was nice. WRIB meeting, and attended the GCC afterwards, which is the CRO consortium. So yeah, it was really, really great to be there.
So moving on, Ravna Veeramatchineni. Ravna, do you want to talk about who you are, what your current role is, background, things like that?
Definitely, John. And you did so good with my last name there, so I'm very proud of that. Hey guys,
practicing.
Yeah. Hey guys, my name is Ratna. I'm the director of mass spec bioanalysis development here at KCAS Bio, sitting at the Kansas City site here. And this is my first time on this podcast, and very excited to be here and to hear as well as talk more about WRIB from last week's experiences. Thanks.
Then we have a guest from our Philadelphia site, Dave Ambrose.
Good morning, everyone. Dave Ambrose here, Associate Director on the operations team. As John mentioned, I'm at the Philadelphia site where we specialize in flow cytometry. Also very happy to be here today with you guys, and it was great to see everyone in NOLA down at WRIB.
Next up, Bryan Parmentier, who we also had on a recent podcast, but this is a different topic today.
Good morning, Bryan Parmentier, Associate Director of the Small Molecule Department, and I do report to Ravna. Been working with the company for about 6 years. Prior to that, about 24 years experience in CRO, mostly centered on mass spectrometry.
And last but not least, Jessica Pham. And Jessica, you're only last on the list because it's just the way it is in front of me.
Yeah, no problem. So I'm Jessica. I've been with KCAS for 3 years in the biopharma LBA space as a scientist.
Myself on mute. So yeah, that we thought we'd at least give everyone a chance to introduce themselves. So, and then the way we set up the agenda is it's give people a chance to talk about the actual presentations that they did. So, Dave, you're the guinea pig. I like to say it was the first time we've ever done this. So, if you want to talk through your presentation, just the reason why we were doing the work, what was the novel aspect, and just the kind of reaction you got from talking to other scientists would be really helpful.
Sure, happy to do it. And although this is my first time on this podcast, the Philadelphia site has one called the Conversational Flow. So I'm just going to plug them really quick, and I have done that. Tune in, it's a great one. But down in New Orleans, I was presenting a poster covering the global harmonization of our spectral cytometers. So specifically, we use Cytec Auroras. They're 5 laser systems. Our primary site, as mentioned previously, is in Philadelphia, and then we have a site in Lyon, France, and a partner lab in Melbourne, Australia. So 6 instruments altogether. And we really took this approach to set up and support fresh whole blood studies. We were using our previously validated 15-color panleukocyte panel. And I think the main highlight, at least for me, and a lot of what was really received well at the conference, was that we invested very heavily in our people and processes. So we had an in-house SME here in Philadelphia who had years of experience on the Auroras, and she was a perfect fit to lead this effort. And she traveled to each site. And so once she got to Lyon or Australia, she was able to perform performance qualifications for all instruments. Once the instruments were deemed good to go, she did on-site staff training just on basic hardware and software stuff, but as well as assay-specific training. We did the assay run using the Strec CD-Chex Plus controls, which is kind of like a dried whole blood sample so that we could compare the same sample across all sites. And then using that data, we were able to do statistical analysis comparing inter-site, inter-instrument, and inter-analyst. The data was super tight. We were very impressed. I think everyone, but our poster was very data heavy down at the conference, and everyone was super, super impressed. CVs were under 2% in most cases. We had a cutoff criteria where if we had less than 100 events, it was deemed out of range low. But even these two populations, which were low, had CVs under 15, which is just incredibly good for this sort of assay. And I guess in general, we are ready and set up to support multi-site global trials, and really looking forward to supporting the industry in that way.
Yeah, Dave, thanks. And the first CRO to have this offering, right? We are the first CRO to have an off-the-shelf spectral flow panel. Is that an accurate statement, Dave?
Yes, sir. Absolutely.
Yeah. We should brag about that one a little bit. I'd love to dive in more about how impressive a 15% CV is. That is amazing. But we're gonna move on, John. But that I want to highlight that the Flow World percent CVs, right, David? If you set up the same tube and run it on the same instrument, right, 3 times, you can see a CV of up to 20%. Let alone if you take that sample and run it across 2 instruments, you can see it. So it's very impressive. I think spectral flow is really revolutionizing flow cytometry. It seems to have much more accuracy, in part. Why, David? Because it takes the whole spectrum, right, instead of gains.
Indeed, completely agree.
So when you say that, do you think flow cytometry wise that the industry is going to move much more towards spectral flow, or is it similar to like mass spec where high res has a really strong niche, but its triple quads are really the workhorse of our teams?
No, I think the industry is heavily moving towards spectral. It just allows you to collect much more information. The data sets are just so much more informative and useful. With that said, and it can even now support primary endpoints for studies. So definitely feel like the industry is heavily moving in that direction.
Yeah, and one other question I was thinking about. Obviously, I mean, I'm a very traditional LCMS approach, but in terms of attendance at WRIB, is there a big flow component, or is you find that it's still early days from that perspective?
I guess maybe we're still a little niche, although we do have a full dedicated specialized workshop every year, which is fabulous. It's super helpful, very informative, getting the industry experts together and hearing what they're doing and what the expectations are. I do feel like, flow cytometry is still kind of in that unregulated space, which WRIB is very much about bringing regulators together with scientists. And we have good guidelines. The document is called H62 that we refer to, but again, they're just guidelines. They're not actual regulatory requirements at this time.
I see Dawn's got something to say.
Yeah, I was just going to kind of follow up on, John, your point around high res, right? Is there any, Dave, in your opinion, negatives or issues or potential, reasons you wouldn't want to do spectral? So, for example, for high res, it's file size, right? It's data processing can be much more complicated. So, kind of to John's question about being the workhorse, do you think spectral can not have any negative disadvantages such that you might want to move to that as a general rule?
I mean, I think the one spot that maybe still is not necessary, but it can absolutely handle it, is if you have a very, very focused question and you want a tight clinical assay with 1 or 2 readouts that's all you're concerned about. It's not required to run that on a spectral instrument. It's more for if you want to be in the 30, 40 parameter space where it really, really becomes helpful. And so if all you have is just a very focused, simple assay, there's no need to put it on a spectral instrument. But again, with that said, it doesn't hurt to. So the data files are a lot bigger because, like Dominic mentioned, we're interpreting the entire spectrum, not just a sliver of the wavelength. So the files are massive, and if your question is pretty simple, it may not be necessary.
Yeah, makes sense.
No, it's a good one, Don, though. But I do think in 10 years, we're going to find ways to compress those files and store them more efficiently. And I do think conventional flow, David, and you can agree or not, is potentially going to go away over time, right? I do think everything, if 10, 15, 20 years from now, you're going to be hard pressed to find a conventional flow, right, Dave?
Completely agree. I don't think I saw a single poster or talk at the conference that had any focus on conventional cytometry. Everything at this point is going towards spectral.
Yeah, and Don, it's a great question you asked, because you don't see that happen in the high-res mass spec space, right?
Yeah, I mean, there's still so much more simplicity and even better sensitivity still on triples. And then the file size, yeah, it's been many years and it's still a problem, right? So be interesting to watch the space.
Yeah, so I mean the amount of data it supports today is a little bit a little more costly. And then the other part that's challenging, David, is just the regression of that data, right? Trying to analyze that data is where I think the future, eventually, as that gets more streamlined, and there's omics and other software out there, right, David?
Yep.
That's when I think as soon as we get a better handle on that, or flow cytometry field gets a little bit more mainstream, that's when I think conventional will go away, almost entirely. I believe it will, because it saves on reagent, right, David? If you have a 2-tube HIV panel,
Yep.
you're using a lot more reagent there, so it's actually much more cost effective to do spectral flow. So,
Sure.
Okay, Dave, any parting thoughts, David, before John moves on to our next guest?
No, I was just going to completely agree with your analysis there. I mean, we still are doing the gating manually, although we've been talking about automatic, unbiased gating now for at least 10 years. It's still not prime time, and so when if you're doing a 30 color panel, you can be generating thousands of reportables, and so to manually gate all those files is still subjective, time consuming, and costly. So agreed that is still a hurdle that we kind of all have to deal with.
David, I'll say that was like that, and oh my God, I'm going to go up like 95. We still had the same problems, although there is that one like TBNK panel that has some auto gating, but it's got problems to it as well, doesn't it?
Yep.
So, John, let's make sure we
Let's
get the
move
rest
on.
of our guests in. Yeah.
Absolutely. Yeah. So next up, Jessica, do you want to talk about what your presentation was, and again, like I say, what the novel aspects are, and the kind of feedback you got? And I thought I see it's quite nice that we've talked about malaria in the news section, and we're coming back to an anti-malarial in your presentation too.
Yeah, of course. I was just thinking that while you were talking about malaria, I was like, wow, this is so relevant. But my talk was over the development and optimization of a quantitative assay for an investigational anti-malaria monoclonal antibody drug candidate in human whole blood using VAMS, which stands for volumetric absorptive microsampling. So the real novel piece here is the VAMS device. In particular, we use Neoterix Mitra devices, and they kind of look like Q-tips. So the cotton portion of this Q-tip, it's a hydrophilic porous material that uses capillary action to absorb whole blood, and it absorbs it in a very precise manner. So Neoterix, the manufacturer, they produce 10, 20, and 30 microliter Mitra devices. And in this case, we use a 20 microliter VAMS device. And so it'll absorb about 20 microliters of human whole blood. And that gets put in a bag sealed with a desiccant to dry. And that dried whole blood on the VAMS device is what we use in the lab for bioanalysis. And this specific poster is looking at developing a PK assay, so a pharmacokinetic assay of that dried whole blood. And with a PK assay, there's a lot of challenges with that because you have to be so accurate and precise. And so we had some challenges getting here with preparing our calibration in QC so that it represents the samples that are coming in, how long our calibration and QCs have to dry, just like how the samples are being dried, and the stability of the VAMS devices, dilution linearity, and the recovery of the analyte. But sorry, go ahead.
No, sorry. I assume that the attraction for the sponsor is that you're hoping for stability at elevated temperatures, because if you're sampling in the field in tropical Africa again,
Yeah.
then that's really the driver here. So you don't have to worry about dry ice and things like that. So I assume you're probably doing quite extensive stability assessments.
Yes, so we're looking at stability at 37 and 45 degrees Celsius because our samples are coming from Africa.
Yeah, and John, they even lack phlebotomists, right? So no tube, right? This is all similar to it's like a mini dried blood spot. Very powerful stuff, as you know, John, right? So it's got not only is it temperature resistant, it has just the cost savings of being able to get samples out of, we'll say resource poor nations. But Don, go ahead. You got a question for us?
Yeah, I was just going to kind of expand on that a little bit and ask about if how the poster was it a comparison to a similar volume not collected through that device, or was it, kind of how did those compare? And then I guess the other question is, did you look at other devices, or, was there a specific reason you picked the Mitra device?
Yeah, so our sponsor, Gates Medical Research Institute, they wanted to use this device in particular because it's so convenient, minimally invasive. All it takes is a finger prick. So we're not drawing 50 or like 100 mils of blood. We just need 20 microliters. So they really wanted to go with this device. So we weren't able to explore any other devices and compare in this case.
Yeah, so their preference, Don, was the Mitra, not anything else.
Yeah,
And,
yeah.
Assume it. Yeah, I
sorry.
assume it's all about ease of use and things like that for the patient. I mean, going back to EBF last November, there was at least one Gates presentation, and obviously logistics is a huge part of their studies, almost more than the sampling from the people. It's what the environment induces on your study and how to make it work in that environment. I mean, it was a fascinating presentation.
Yeah, I mean, it's an evolving field, right? Where everybody's doing microsampling both for a small volume, but also for, like you said, these places that are hard to get samples out of. But, Dom, you kind of mentioned dried blood spots. It's interesting to see how the field is
Hmm.
evolved and moved into different directions. So I think
Yeah.
stay tuned.
And Jessica, you did encounter some challenges. You touched on them, how to get the calibrators and QCs, or excuse me, the standards and QCs set up accordingly, right? That took some artwork, didn't it, Jessica? Right, to get that so that it mimicked the sample. Is that right?
Yes, correct. It took almost 2 years to kind of troubleshoot all of these bumps in the road to get here.
Mm, wow. I'm sure we could dive into more on what that all entailed. But Don did ask, so you guys didn't, Don's first question was you didn't actually evaluate versus like a standard serum plasma sample, right? Or did you look at that as part of this?
So we do have another study also with Gates Medical Research Institute that explores just serum. And so
Okay.
we kind of use that to help correlate, because we do use serum in this study to spike our analyte in and then further spike our whole blood.
Okay.
Dip our VAMs in that, let it dry, and then work from there.
All right. Don, did that answer your question there? So they did have some way of kind of comparing it to.
I think again that's historically what I've seen as a comparator is you always compare these newer devices to collecting it, in the let's call it the traditional way.
Yeah.
Um,
Yeah.
and
And
usually those are the datas that help with, a WRIB, right? A regulatory kind of acceptance, showing that cross comparison is always nice.
and maybe one last question for Jessica here. I don't know if you know the answer. I don't know if anybody knows the answer, but I think are we one of the first to validate this? Is that a PK method for biologic, or have there been others that have done that? And maybe Don, you might know. I don't know. Feel like in the APS community, there's been people trying to do this.
I think, I mean, again, if Jessica's been working on it for 2 years, it could be we were one of the early ones, but I also think, like you said, I think there are several people that have been working on different devices, whether it's
Okay.
Mitra or Tasso or, even dried blood spots, right? Just different types of
Yeah.
validation.
Well, it's certainly one of the few, right? I think this is a really hot topic. Hot topic, excuse me. There's a whole AAPS group dedicated to microsampling, so it is something that the field's interested in. And John, it ties in nicely into what we had with our patient-centric sampling from a few weeks ago, right? With
That's exactly what I was going to say.
yeah,
Yeah, because I mean, it's, and I think, yeah, I don't know how much that was biologicals versus small molecules, but yeah, it does point to how much interest there is in this area.
hmm
Just because it's all about making it better for the patients.
yeah, which is still fat. Like even as I sit here as an industry, we've just 50 years collected blood the same way, right? And so, but why are we doing that? But
Sure.
I don't know if we have anything else for Jessica, but I think we can move on to Bryan.
I think let's move on. So our next guest up, Bryan Parmentier. Bryan, do you want to talk about your presentation background and what's the novel piece of it and reaction?
Sure, John. Hi, so my project was the determination of cisplatin in human plasma, and we had observed significant matrix effects during development. So the poster was focusing on using the novel idea of a hemoglobin reagent to overcome those matrix effects. Um, Overall, during development at KCAS Bio, we work on a lot of troubleshooting of matrix effects. We see them very often, but this was a significant case where if you had a hemolyzed sample, we were seeing accuracy down to about 10%, where our accuracy needs to be between 85 and 115%. Ordinarily we can make simple adjustments to our methods, diluting the matrix, or revising the analytical method to get around interferences. But in this case, we had to take a deeper look. So we had scanned in single ion mode some of our samples within our batch that were showing non-precision. And what we noticed that there was an ion 616 that was very prominent, but it was variable in between samples, especially in hemolyzed samples that you could visually see some hemolysis, a reddish or orange tinge. That ion was higher. But it was also observed in all plasma samples that we looked at varying degrees. Um, at that time, we had also come across in the group of scientists that worked on this project a paper where that same ion, in coincidence, was seen, and it was a paper investigating patients that were undergoing cisplatin treatment and that had a trend of an anemia. So it wasn't an assay paper, it was just that they were investigating the patients and they had noticed that ion. So they had associated that in vivo with an increase in cisplatin causing a decrease in hemoglobin. Thus, with that decrease in hemoglobin, you have a decrease in iron and ultimately anemia, right? So we took that in vivo phenomenon and we applied it to our in vitro assay. We worked with whole blood at first, treating our whole assay. As, if you can imagine, it's a plasma assay, and then we're treating it with whole blood. And when we did that, we were able to equilibrate the methods that all samples, the cisplatin levels could be accurately recovered within criteria. So We carried on and we optimized the method further so that day to day we could have a reagent to use of hemoglobin. We had found a lyophilized hemoglobin through a vendor, and we optimized that to a concentration about 10 times less than the in vivo concentration of hemoglobin in humans. And it was easy to work with, and all of our data was acceptable in the project, and ultimately it was validated. So it was a really neat troubleshooting project, and we're going on to use that project in cancer study.
So I look at cisplatin structure, and it's a very simple structure. So was there any issues in terms of ionizing, and what fragment did you follow? I mean, it's basically what a platinum surrounded by two chlorines and two ammoniums.
We follow the fragment of 492 with a product ion of 114. Yes, and it is a derivatization method.
Okay.
So you take that into account. That is out there in literature. But overall, what we see time to time is that some of the validated methods that claim to be validated in literature still have issues or assessments that weren't done at a certain time because they weren't required. In this case, if you look at hemoglobin itself, hemoglobin is known to degrade both through temperature, storage duration, and handling. So that is a potential cause for why you can see varying degrees of hemoglobin even in plasma matrix that looks good to the eye where you're not observing hemolysis. So in our case, we were using fresh whole blood and fresh plasma to optimize this assay, and we noticed that depending on the plasma that we used, if you used aged plasma, we were seeing lower levels of hemoglobin, and we were seeing data that was more acceptable. So that's some of the underlying, things that can happen in these assays if you're not taking a deep enough look at the development, and you're not using fresh reagents and fresh matrix. You can potentially develop an assay that does meet criteria, but then when it comes to study samples, you may have a different outcome, and you may see these interferences come to fruition.
Yeah, I've definitely seen assays in the past where age of matrix was critical. I'd worked on one years ago where there's a parent and metabolite in rat plasma, and the parent had a stable label internal standard, and the metabolite didn't. But if we used old plasma, which we hadn't realised at the time until we're in the middle of study samples, the metabolite was fine, but the parent failed, even though it was the one with the stable label internal standard. And as we did troubleshooting, it was traced back to the age of the plasma that we'd used for our standards and QCs. So yeah, you go through that once and you learn your lesson, and it becomes something you're alert to after that.
Yeah. So as somebody who's on the biologic side here, I got a quick kind of question for you, Bryan. So if I understand this, you're maybe it's two questions. One, if the sample's visually hemolyzed, or is it just in general if you see a hemolyzed sample, are you adding like exogenous hemoglobin, or is it to all samples you're proactively adding it? Do you understand my question?
Yes, I understand.
The solution, right? The solution is to add. See, it's a little counterintuitive. You think of a hemolyzed sample, you're adding more hemoglobin, and what's the maybe just outline how it works. Sorry to ask two questions at once there.
So your latter statement was that is it added to the whole assay plate? And yes, it was. So it's added to every sample.
Okay.
Because you have to remember that hemoglobin will be present at some level in all plasma samples, regardless if you can visually see the hemolysis.
Okay.
We all have an underlying baseline of hemoglobin in plasma. That's from the lysis of red cells. So what we had to do is optimize the assay, because any plasma that you had taken, depending on the amount of the hemoglobin present, would automatically conjugate with cisplatin. So depending on the degree of hemoglobin present in the plasma, such and such cisplatin would be taken in, and it would cause trouble with the assay when you're trying to match it against standards in QC. So overall, you're just going to have a lot of variability.
And that's how you normalize it, right, Brian? You're just driving everything to be hemoglobin conjugated, essentially. All the cisplatin gets hemoglobin conjugated in the method, right?
Yes, and the neat thing is, though, once you do that, you're able to recover properly cisplatin through the extraction. So it's just that upfront work that has to be done, but the extraction itself.
Very clever. Is this? That was. It's Brian. Did a paper help you? Is this really? Are we the first or no? This was a little bit known. You just adapted it to cisplatin. Help me understand how we kind of got to the solution.
The solution that I had come up with was just the in vivo observation that patients on cisplatin had a trend of anemia.
Okay.
So I took that idea and made an assay out of it in vitro.
Yeah. So are we the first to add like exogenous hemoglobin? Oh, we are. Wow, that's
and so the plan is to publish this beyond the presentation at WRIB. So
I got one more question. Is so cisplatin is look? Forgive me, right? I know enough to be dangerous about small molecules, but there are many other small molecules that are in the cisplatin family. Is this applicable to those small molecules as well?
I'm not sure.
Okay. Well, I think Dominic, the platinum-based anti-cancer drugs are all quite variable in structure. Because we also did some work on carboplatin, which is much more complex around the platinum. So I think I probably think there's more protection of the platinum within that molecule. I'm going out on a limb here, but cisplatin really is a very simple structure. There's not much to it at all. So I wonder if it's more prone to this kind of effect.
Because of I got you. Because of all right, I'll put myself back on mute.
I think we're good. I don't. Is there anything more you want to add, Brian, or we could move then on to the just discussion of people's overall feedback from the WRIB meeting?
Just quickly, I could give my feedback now from the conference. The conference went well. There were a lot of scientists taking pictures of the poster. I did actually meet one scientist that had worked on cisplatin, and the assay wasn't working well and ultimately stopped working. So they had trouble too on LC-MS/MS. So they were very interested in the poster.
Wow, that's awesome, Brian, right? That's got to make you feel good. You solved a problem for someone. That's amazing, right? Yeah. Any other parting thoughts, Brian? Sorry to cut you off.
No, thank you.
Last thoughts there. Did you get some good food? Did you enjoy New Orleans? No.
Yes, the food was very good. Thank you.
All right, we're gonna set our sights on somebody else now, John. Right? I think it's a general discussion. I see Don. Don, why don't you tell us why don't we jump right into you, Don? You want to?
Yeah. So I think, WRIB I've been going to for, I don't know, 12, 13 years, for a long time. And I think it was, similar to past. I think a couple notable differences that I think, obviously the FDA's presence, or the regulatory presence, was more virtual this year. We've seen that in the past with, COVID years, et cetera. But I thought that was a notable difference. The other thing that is probably really good to point out is, really, this is a meeting to bring together different parts of our industry such that you get some alignment. There's a lot of what I'd call consortiums or consortia where we try to come out with guidances from this meeting. There's always proceedings from this meeting. Anybody that participates in the working dinners or the talks specifically, whether they're specialized specialized or main day talks, there's long dinners, there's papers that come out. So it is a very pivotal key meeting to share guidance and consensus, let's say, across the industry. So that was always the same. One of the big topics that I want to bring up here that is, I think, still to be determined is the this year's kind of recommendation or guidance from the FDA on biomarkers, and that we now should be using the ICH M10 as a starting point. That is a controversial topic, let's say, at the least, because if everybody remembers, the ICH M10 in the past has specifically excluded biomarkers, and now all of a sudden they kind of pivoted, did an about face, and said, oh, we're going to kind of include them here. And so I think that's stay tuned, because there's a lot of different groups, either at WRIB, the GCC had a lengthy discussion at all the CROs. There's, AAPS focus groups that have specific opinions about it. The EBF has a specific opinion about it. I think fundamentally it's no different than we've always done good science, but the devil's in the details of the interpretation of that, and there'll be specific implications with respect to, like, let's say for KCAS Bio, right, and other companies like CROs or sponsors, whether we call things validated, whether we call them qualified, whether that means they're QA reviewed. There's a whole lot of conversations going up there, and I don't think there was any direct consensus completely, except for that we feel like it didn't really change anything, but it's changed everything. So it's kind of one of those circular things that some people don't think it does anything, other people think it's a complete about face. So I found that very, very interesting. I don't know anybody else's thoughts on that particular topic.
Yeah, if anybody has thoughts, please share. But this is a whole another podcast on, right?
Yeah, for sure, 100%. But it definitely was the first place I had been to since that guidance came out, where it was, a conference where everybody was discussing it pretty vocally.
Yeah, no, Rathma, please share your thoughts.
Yeah, no, I think that's a good segue into what I heard as well. I got the opportunity to sit on a lot of talks and presentations, and got to talking to a lot of other industry leaders in the different topics that are active right now. And one of the main topics that every one of us were discussing time and time again, each day we were there, is biomarker space, whether it be the multiplexing of the biomarkers of how you perceive it for the M10 guidance or not. Same thing with respect to how these would dive in, when you dive into tissue homogenizations and tissue biomarker assays, how well can you adopt these kind of guidances into tissues, and looking at super low levels of biomarkers of how this guidance can be applied to that versus not. There were like several conversations on this topic on many different levels for biomarker bioanalysis that came up during all of these conversations as well. I think I would say primary topic, it was biomarker quantitation through all these different arenas, outlook into that, I think.
Maybe just, sorry, go ahead, Ratha.
No, go ahead, Dom.
Yeah, I was just going to say, maybe to round it out, and others can chime in, and, like Dom said, I think this could be its own podcast and topic, but there's also a little bit of a semantic argument, like what do you call it, how do you do it, if you're still doing the same things. But there's also a big, what I noticed, a big difference of opinion, let's call it, on calling a biomarker just kind of like an endogenous PK assay. So people that are used to PK assays and think about the M10 just say, well, it's just an endogenous presence you have to worry about, and everything else is the same. And I actually feel like I've said that in years past. But as you learn more about the other perspective from the biomarker community, and specifically like the global consortium, the GBC or the APS leadership, they 100% don't have that opinion, because it's all about this reference standard, and you don't really have a true reference standard. You have post translational modifications and all of these things that really make a huge difference, and you don't have something you're really spiking in. You have something that's maybe similar or could behave, you're using as a surrogate. So there's could be semantic arguments, but very clear, strong opinions about kind of some of that. So it is very interesting topics I found.
It really is. I think end of the day, what we are also observing in the biomarker space is something that's endogenously present or produced in especially these different biomarkers, actually both in the small molecule and the large molecule realm that we are going after. What we find is when we have a recombinant or a synthetic molecule, sometimes how they behave in matrix upon spiking is definitely, and we have seen this in our hands as well at KCAS Bio, not behaving the same way as a endogenously present bound interacting molecule. So how you would then align yourself with that M10 guidance is very interesting, and it can be a case by case basis, but there definitely are a few outliers that we definitely need to watch out for if we sign ourselves up to be able to matching up with the guidance that's out there.
Yeah, so maybe John and Dom, we have a nice topic for sometime in the future, but maybe we give it a little time to settle.
We're gonna have some internal.
I think that's probably the case. Yeah.
Yeah, we're gonna have some internal discussions. But Don, you said it nice. Like, some people feel like it has no impact. I probably fall in that bucket. And other things think it's like we're gonna have to revolutionize how we're doing biomarkers. Everything in between, right? There's everything in between. So let's maybe Ratan, do you have any other thoughts about WRIB before I turn my sights on Jessica?
Dave,
I have be ready, Dave. You're next. Go ahead, Ratan.
Yeah, couple of things. I definitely added it to my list of things that as I sit into these different talks. The main topic, that the second one that I felt like was most talked about during this last week when we are there, during the talks as well as offline talks as well, is a lot about interference RNAs as well as antisense oligonucleotide bioanalysis. There were several topics on lipid conjugated version of the interfering RNA bioanalysis. And the prime topic, which as we know it, KCAS Bio is working on a few projects, if not a lot, on metabolites within this RNA therapeutic analysis. That was another topic that was discussed in a couple of the talks. That was very interesting to see. Last year, I don't believe we had any conversations on metabolites within RNA therapeutics. This year, I feel like I saw some more of that come out. We also had a couple of talks about hybridization ELISA combined with the LC-MS work. That's something that's starting to show up more in some of the conversations and talks as well. And then there were a couple of conversations about oligonucleotide conjugated antibodies and how do we quantify the payload, whether it's a total antisense or free antisense assay, why we are doing what we are doing, that sort of a conversation. So it was very stimulating and interesting. What I found through all of those conversations and talks is we are in the forefront of applying majority of those ideas to the current work that we have here at the company. So I felt really good about listening to all of that. The third topic that came to be, which we are already doing it in a really good way, is the ADC conjugate analysis. There were several topics about best practices, and then there were a couple of talks about the 1.5 assay format for the ADCs, which we have validated several assays at KCAS Bio using this format. So that was really good to see that people are starting to talk about it more. And we actually have a poster coming up in the upcoming AAPS NBC conference as well with the 1.5 assay format. The other interesting thing that I found is a topic that we discussed on these bicycle drug conjugates that sit right in between the antibody as well as the small molecule area, where it meets the requirements of small molecule, but it behaves like a large molecule. So that was a very interesting topic to sit in and listen on one of those talks. A couple of random things that I felt like were very interesting to listen in, as well as see that there are several others out in the industry that are in a similar thought process as us, is There was a topic about the isotyping and semi-quantitation of the ADA assays using hybrid LCMS approach. We did some of that a couple of years ago in a very qualitative manner, but that was really good to see from a pharma company that was presenting that. And then there were a couple of talks about non-specific binding and extraction recovery. Although it's an old topic, they want to revisit it as they're looking at it from a new light, etc. So that was a really interesting conversation. And then some bottom-up and top-down glycan analysis, which we are starting to see, similar to what Don was describing earlier about the post-translational modifications and things like that. So these were, several interesting, stimulating conversations. But one thing that stood out for me again and again is a lot of these topics we are already touching on, we are already exposing ourselves on, experiencing on. So that was really good to see for me.
So rather than the non-specific binding, what sort of matrices were they digging into? That was it urine or more things beyond that? CSF? What kind of things?
Yeah, exactly. Urine, CSF, these sort of things that doesn't have a lot of proteins present within the matrix. So therefore, you're seeing a lot of this. Unfortunately, these non-specific binding that gets into inconsistent recovery, and how do you adopt to fix those different scenarios?
Yeah, I think a couple of things too there. Ratha covered like about 100 topics there, and several of them could be their own podcast as well. But a couple of things maybe near and dear to my heart as well is one, the ADCs, right? We have actually Dom and I have a X-Talks webinar coming up in a couple days on some of that. So I think that's really interesting. I like to say, and I think this is an accurate statement, that KCAS Bio really did coin that term 1.5 plex assay. And it is interesting and nice to see other people start to understand and use that and understand the power, of doing that type of thing. So I think that's pretty cool. And as Rothan said, I think the oligos was a very much bigger agenda item or part of this conference than in past. And again, similar, just reiterating maybe all the different technologies that can and need to be used, whether it's hybridization ELISA or high res or triple quad or mass spec or whatever. Lots of different PCR, et cetera. Lots of different approaches that are needed in that field. So it was a very nice conference on the science end, and I really appreciated that.
Yeah, Don, it's great to see that we're staying ahead of things. The 1.5 is definitely, I think you can assure that 1.5 plex for protein mass spec, hybrid mass spec, that we own that, I think, or at least to some degree you do, and Ratna. And then I'll just say, hey, it's not a surprise that oligos are now at the forefront. The GLP-1 space, as well as people are starting to put, taking a biologic and throwing some sort of siRNA on it is all the rage. So it's comforting to know that we stay ahead of the curve. That's why we call ourselves an innovative CRO. We're running short a little bit of time, but I want to give Jessica a couple minutes and let's try to get Dave in real fast. So Jessica, why don't you share a few thoughts about your first WRIB experience and what you thought was some of the highlights?
Yeah, so it was my first WRIB, so I didn't really have any expectations of it, but I thought it was really great. I think the best part about it was just talking to everyone that comes from different backgrounds, different expertise, and seeing their perspective on science. So talking to marketing, commercial, the regulators, even just the scientific advising group. And even talking to other companies and what they do at their labs, what are some similarities. There were a lot of people that came to my poster saying that they work with whole blood and find the same issues that I'm finding, and then we're sharing our experiences, challenges, successes, and giving each other ideas on what we should do next, how to combat all of these issues that we're seeing. So I thought it was great.
Yeah, the camaraderie, you bet. And it's electric. I call it the Super Bowl, and now you know why. David, go ahead and quickly share some parting thoughts for us as we wrap up here.
Completely agree about it being the Super Bowl. It is my favorite conference. It's the most relevant conference, or at least for flow cytometry. We're able to get together with the industry experts and make sure that we have alignment and are able to support them. that our practices match up with what they're doing. And so, it was always good to see that our company is right on target there. And it was a great time in New Orleans, able to hear some great music during Jazz Fest, and the food is fabulous as always.
Yeah. All right, John, any parting thoughts from you? I don't have much to add. I think everybody covered everything for me. I unfortunately had to leave a little early to go see the Frozen Four, which, by the way, Western Michigan Broncos won. Hail to the Broncos. Okay, sorry. That's it for the Frozen Four. If that's the hockey tournament, I did get to see the NCAA hockey finals, which was amazing. But John, some parting thoughts for us.
I don't have much to add. I mean, I'm sitting a little bit on the sidelines. I've never been to WRIB, but I have had the benefit of doing EBF, which is like the European equivalent, as I understand it. So
it's different.
And I would definitely say I'd rather do that, because as good as the food is in New Orleans, I think Barcelona.
Oh yeah, it can't be. And you know what? The strange thing is that they're quite kind of different in terms of size and structure, but you get the same outcome. The camaraderie.
Yeah.
Just think about the depth of what we talked about: biologics, biomarkers, small molecules, flow cytometry. I mean, it has everything for
Yeah.
If you're a scientist and a nerdy one, WRIB and EBF are both they're just great. They're fantastic.
Yeah, they're both excellent meetings. So yeah, that's all I wanted to add. And yes, thanks very much everyone for joining us. Like I say, we weren't quite sure what we were letting ourselves in for, but we thought we'd try and do something a bit different. I think this has been great.
Yeah, and no hot seat.
Thank you.
You guys are all out of the hot seat because we're running short of time. But John, I'll see you in the funny pages.